My name is Pete Heintzman. I'm in the Paleogenomics lab here at the University of California, Santa Cruz. I'm a postdoctoral researcher in ecology and evolutionary biology, specifically using ancient DNA and ancient genomes to explore how organisms from the Arctic moved through time during the last ice age and how they moved around. So part of the reason that we want to explore how populations move and how they respond to past episodes of climatic change during the last ice age is to use that to help infer how species may respond to future human-driven climatic and environmental changes. My father is a scientist. He's trained as a scientist. He didn't actually go into science, so he did an undergrad degree in chemistry. So I kind of had the science from him a little bit, and I always enjoyed doing it at school. So I enjoyed science and maths at school and had some very good teachers. This was back in England. And from that, I decided to pursue an undergrad degree in biology. But I was also fascinated with, I was also good at and enjoyed chemistry and geology. So my last years at high school, I did biology, geology, and chemistry. But pursued biology as my undergrad degree, but then I did a master's degree where I kind of went back to the geology and actually did a master's in paleobiology, and then went to Royal Holloway, and that's where I did my PhD. And then immediately after I finished that, I came here, and I've been here for the last 2 years. I'm just starting my third year here. It's incredibly variable what I do on a day-to-day basis. So if I'm doing lab work, it could consist of a day doing DNA extraction, so grinding up bones, adding various chemicals to them to really isolate the DNA. And then there are other techniques we use which convert that extracted DNA into DNA that we can put on the DNA sequencer, and then we can do the sequencing. So some days I might be just in the laboratory all day doing what we term wet lab stuff. Other times I'll be doing dry lab stuff, which is more of the computational side of things, which consists of tidying up sequence data, assembling the data against another genome, and then running various analyses on it. And then there are the other parts of science where you have to analyze the data and then write things up for publication. But some days it's completely different, so I might go off to conferences where I'm presenting the work. The important thing at the moment is the whole field is going through a lot of methodological developments, which has actually allowed us to be able to reconstruct paleogenomes. And a lot of stuff has recently, in the last 2 or 3 years, been published, especially on humans and a few other well-known animals like horses, really exploring some of these questions about how humans and hominins, ancient archaic human-like animals, evolved and how they relate to modern humans, and also this idea that some archaic hominins like Neanderthals actually interbred with humans. So what really motivates me to want to come in the morning is the fact that some days you make a discovery that no one else has made. It doesn't happen every day. In fact, it happens on very few days, but you have no idea what day that's going to happen. But when it happens, it's absolutely amazing, because you'll be analyzing the data, and then something will be thrown up that really makes you think, what's going on there? That's weird. 90% of the time it's because you made a mistake and it's not actually that interesting, but then that 10% of the time where it is really interesting, it's fantastic, and then you're like, that's great, this is why I come in, this is what it's all about, this is why I do science.